Oh yeah, so back to this yesterday. So we raised $1 million from families and are giving out $652,000. And so to these 5 projects. So we've got 4 of the speakers here today. So you guys have, well, it's only 14 minutes now. So you guys have less than 5 minutes each, maybe 4 minutes each. Okay, so Dr. Frankel is not able to present. So we can move on to the next one. So Dr. Coombs.
Yeah, I should point out that that may be a familiar title to Dr. Lal, because it's actually your project, I think. So that's not what we're doing. Did I send you a couple of slides? Are they on here?
Yeah,
they are. Oh yeah.
The right title should be there.
Let's see. Yeah, okay. I wrote this one. Yeah, this is definitely it. So AMP-receptor positive allosteric modulators as precision therapies for Greer loss of function. So I will be a little bit quick. I won't go into my hilarious analogy about being hungover and taking paracetamol as a non-precision therapy, and instead grabbing myself by the lapel the evening before, slapping myself a couple of times, saying, "You're not 22 anymore. Don't do it." Okay, so anyway, I'm glad I didn't do that. But so precision therapy is tailored therapy that's tailored to the disease of an individual, and in terms of genetic disorders, we think of drugs that can directly counteract the effects of a variant. Now, in terms of Greer disorder, and depending on who you speak to, these are things that are really working very well, or potentially there are people who want a bit more clinical data before they're prepared to actually say all this, but nonetheless. For brain gain of function, we have a couple of NMDA receptor inhibitors, memantine and ridipridil, also adamantine as well as kind of on this list. So if you have overactive NMDA receptors, you can reduce the function. For loss of function, L-serine is this, still a bit controversial, but the data is looking good. Again, you can upregulate underactive NMDA receptors. For Greer, we've got if you have gain of function, we have perampanel, which is an inhibitor. But for Greer loss of function, at the moment, there are no clinically approved drugs that directly act to upregulate AMP-receptor responses. And this is despite the fact that many of these have been developed. Lots of them have been in clinical trials. They're safe, but for whatever they've been trialed against, unfortunately, they've never actually got over the line to be approved. And yeah, I won't go into the mechanisms. Let's kind of skip over. And so, what the aims of our project are to first of all, to understand as much as we can about the loss of function variants you see in the Greer genes. So we know that there are, in theory and in practice, what we're seeing in our preliminary data, there are many different ways of breaking an AMP receptor. So we know they bind glutamate and they open an ion channel. So some of them don't even bind glutamate. There's something wrong with the ligand binding units. And others of them bind glutamate, but that's not transmitted to the pore, so it doesn't open the channel. And other times you can bind glutamate, you can open the pore, but then it desensitizes and closes very, very quickly. So we want to work out exactly what we can, find out exactly what can at specific variants, and maybe even relate that to symptoms. And then I guess the meat of it is to then look at a couple of PAMs that are proven safe and have been pretty close to being approved at various points. One is VIIB, I think the Pfizer currently own this, and the CX-1739 that there's Respiralex are working. And they seem to have a slightly different mechanism of action. There's a high impact and a low impact one that may impact the safety margins and exactly how much use they are. And what we want to do is to try and look at all of these variants we characterized here, see which kind of PAM is more suitable for which kind of variant. We're also going to do a bit of structural analysis, looking at whether any of these variants affect the structure of the receptor, and also look at the basis of this low impact modulation, because that might be very important for safety margins kind of going forward. So these are the 3 main strands. And ultimately what we're hoping to do is to facilitate future clinical opportunities for trials of these drugs that could ultimately be used to treat patients with Greer disorder. And I'm about to receive a jiffy bag with one of these compounds when I meet with Arnold Lipper in a few hours' time, and that will be going back to the lab, and we can kind of get started on these experiments. So yeah, that's the project. Carry on in there.
Okay. Next up, so Dr. Kalev, who's working on a project, I guess, with assistance from Dr. Ramsey. Oh no, it's not working.
Hi, so my name is Maggie Caleb. I'm a hematologist scientist at the University of Auckland. Thank you for having me here. This project is in collaboration with Dr. Amy Ramsey from the University of Toronto. And really the title says it all. We are looking into blood cells as the source of potential biomarkers for brain disorders. So the project builds on the observation that energy receptors are expressed in several types of blood cells. Therefore, we would like to check if there are changes in blood cell indices in GRIN patients. So we would like to work with the GRIN registry to create opportunities for families to upload blood results and provide us with some pertinent clinical information to help us interpret those blood changes. That would be through a questionnaire. So ideally, the easiest way to interpret the blood results is when they are tested at a steady state, but we know these patients have a lot of problems. So in a way, it would be actually better if we had multiple tests from a particular patient. We are interested in to know acute problems that people have in that week before the testing. So generalized seizures, infection, trauma, bleeding. The questionnaire would supply the list of those. We would ask you to tick, and if any apply, just provide a little bit more information about it. The same with the chronic diseases. We want to know if children have chronic problems with the heart, lung, bowel, kidney, liver functions, or chronic blood diseases, what drugs they are on, their current weight, and why the particular blood test was done. So really the aim of this study is to check if there are any unexpected blood changes that we can detect in routine blood count parameters that potentially could be useful for monitoring these patients moving forward. And the other component of the project takes advantage of the mouse models of the green gene mutations that Amy works with. So we would like to characterize some phenotypic characteristics of red cells, platelets, and white cells in these mice, and determine the pattern of the stress response in the endoplasmic reticulum in selected cells. So just to explain it briefly, when I get stressed, I just get chaotic and anything can happen. But at a cellular level, the stress response is actually quite a highly regulated, organized, and progressive process that happens in relative proportion to the insult and with the aim to restore function, or if that's not possible, to restrain it and prevent cell death. So we think that ER stress will develop in the context of green mutations because cells need to deal with the load of the mutated protein and potentially calcium imbalance. So we want to track through high throughput gene expression sequencing or proteomic studies changes in these responses to try and characterize the pattern of response that happens in cells. So our hypothesis is that by determining that pattern, by knowing that pattern, that could give us a clue about the severity of the insight and the cellular level. And potentially that could help reflect the severity of disease, so potentially help with prognostication and monitoring of treatments in the future. So that's really all. Thank you. We appreciate your funding.
Okay, next, Dr. Boyd from McGill University.
Okay, can everybody hear me? So maybe I can just thank GRICon for funding our project. That's a good thing in the first place. So thanks to you guys. Thanks for doing that. It's a good thing. So maybe I can just start by saying, first of all, this is the first time I've come to this meeting, and my lab came to GRIN disorders actually from an unexpected place. We were looking at Fragile X syndrome, which you might think what's Fragile X got to do with grin disorders? But in fact, you know, I actually teach medical students, and if you look at the clinical profiles of kids with Fragile X, grin disorders, SYNGAP, there's a whole bunch of neurodevelopmental disorders out there. Then they've got a lot of core features, clinical features, which are very similar. And so we were chatting about this in the lab. We said, well, we're looking at Fragile X, but actually maybe a lot of the things that we're looking at are probably common to these. And because our lab's got a long-held expertise in glutamatergic transmission and glutamate receptors, we sort of honed in on glutamate receptors. And then the other thing, of course, if you went to Mike Fraglia's talk this morning about Fragile X, he sort of said it's all bust. We tried, we failed. And so we concluded that there was nothing wrong with what those researchers were doing. It was all perfectly good science. You heard him talk about Mark Baird, and he's absolutely a great scientist. So it's not like Mark Baird got it wrong. We just thought that maybe what's gone wrong is that there's some new biology you have to discover first, and it's only then you actually understand the reasoning for why the drugs are not working. And so that's how we approached it. And so we actually found new bits of biology, and that's where our therapeutic approach takes place. So hopefully this is going to work. So if you're an undergraduate student at McGill and at UCL, where Ian was and I was a long, long time ago, then what they tell you is that NMD receptors, which is what GRIN is all based around, then this is the good guy in the brain. It's an NMD receptor that couples up to this kinase called CAM kinase 2, and that's been tied into learning and memory mechanisms, all the things that happen in development. And then the bad boy of the glutamate receptor world is this NMD receptor that couples up to neuronal nitric oxide synthase. And you'll find lots of papers of people tying that into the damage that occurs in stroke. You know, one of the biggest problems in stroke isn't that you get the stroke, it's all the cell death that happens afterwards. And who's the culprit for that? It's supposed to be nitric oxide and NMD receptors that drive all of that and all this second messenger pathways. But what we found is that when we started to look at cells in the brain, there's only some of them, but they're all across the brain, that actually couple up to NMD receptors. There's actually a biochemical coupling. It doesn't look like it's here. When they couple up to neural nitric oxide synthase, they produce nitric oxide, and you probably haven't heard about nitric oxide unless you went to the dentist when you were a kid, and they usually put you under anesthetic with nitric oxide. And that's because that works, because nitric oxide will put you to sleep, right? But it does the same in the brain. It's involved in sleep mechanisms in the brain. It's involved in learning and memory mechanisms. And so it turns out it's not so pathological. It's actually an important signaling molecule in physiology, and that's what we found. And so what we found is it's this part of the NMD receptor, not this one, that's defective in Fragile X. And what's the defect? It's a hypofunction in NMD receptors, exactly what you find in GRIN disorders, right? And the NMD receptor, I haven't shown all the things here, but it basically acts as a conductor in an orchestra, and it regulates a whole bunch of different processes, including local blood flow into the brain region where you get a lot of activity, neural activity. And in Fragile X, that is all gone. However, because we know about the pharmacology of some of the signaling pathways here, so we don't target the NMD receptor, we actually target all the things under here, then you can completely recover all of the defects we find in Fragile X. So having understood that, and this was all happening just as COVID kicked in, and I was listening to Keith's podcast on CBC, I'm in Canada, and I thought, oh my God, this might tie directly into GRIN disorder. So we got in touch. McGill let us work, very kindly let us work while everybody else was out. And so we developed this whole project. McGill put a patent forward for it because they felt there was IP there. And we developed a spin-off company because we realized that McGill wouldn't have any money to run clinical trials and things like that. So that's where we are with the Fragile X. And so the reason we wrote to GRIN to try and raise a little bit of money is that the drug companies who are going to spend all the money to do these clinical trials, I made a deal with them. If I can convince you that you can get the same effect in GRIN disorders, then they will include patients with GRIN disorders, particularly the hypofunction kids, right? So just to convince you a little bit more about why we think nitric oxide signaling, which you've never thought about as being involved in a disease like this, then the thing to think about is we did this very, very simple thing. And this table can go on for pages and pages. We've done it for literally hundreds of behaviors. So here is a bunch of behaviors you can do in a mouse. You can do in a mouse, or you can do in a patient. And we looked at mice that we've got. So these are Fragile X mice. So they're missing this gene that makes FMRP, right? And then we get another mouse that's missing if I go back here, it's missing the gene that makes nitric oxide and causes cyclic GMP signaling. But if you look at the two of these, and then you compare it with patients, what you find is you look at all these different kinds of behaviors that other people have done, not that we have done. And what you see is an incredible correlation between when it's very hyperactive in patients, you see that in the mice, but you also see it in mice that don't have high levels of nitric oxide. And then something that come up in the session, you know, the session we had right at the beginning yesterday with the 4 different groups of people who were talking about different things, aspects of having brain disorders. One of the things that popped up was aggression, and it should be here somewhere, right? So aggression is elevated, of course, in almost all kids with neurodevelopmental disorders. And what's well known to regulate that is nitric oxide. And so that was like the light went off, right? So if you get low levels of regulation of nitric oxide, then it boosts it up. So you get more aggressive. So the drug that we've got, actually it's a drug combination therapy that we have, we think can elevate all of these. We've actually started to go through some of these. And in every case, we seem to be able to correct things. So what we want to do, so what we want to do is now try and get someone who's got real money, not what McGill's got, and to convince them to run a clinical trial. So the money that we get from GRIN, I think is going to be very important to convince these big pharma companies to mitigate, they feel that they've mitigated the risk and therefore, maybe run the clinical trial. So I just want to finish on one slide. Just want to thank everybody in the lab that's involved in doing this. We've had collaborations with other people as well. And thank you for your attention. And thanks for the funding. It's the more thing.
Yeah, don't thank me. Thank all of the other all the people that have raised money for us. Finally, I know we're a little bit over time, but Dr. Dennis Lau, thank you.
First of all, thank you very much for the opportunity, and I can guarantee you that we will work very hard to move things forward. So I can make it very brief. So imagine there would be a therapy for any of the GRI genes today. Let's call it GRIC3, and there would be a therapy that only works for mixed function, right? So because today we classify arbitrarily almost all the variants into mixed gain and loss of function, which is not what's happening in cells. But this is what helps us as humans to explain the function more or less, and where we have strong hope that therapies could be binned also in these groups and they show some efficacy in patients. So there's good reason to believe that this simple framework of gain, mixed, and loss is actually working. However, if there would be a therapy now again for GRIC3 loss of function, or call it GRIN2A gain of function, and we would ask, where do we have most patients with this specific type of variant? There would only be very few people who have a chance to answer, or to look at a variant and have some idea what it could be, and there's no confidence measure. And most providers all over the world which recruit individuals have no clue how to, classify a variant. And so the beautiful opportunity is that all the money you have invested over the last years went actually went actually very far for the GRIN genes, at least, where we have built already a very large resource. And I mean, Dr. Trenelis and many other scientists around the world, where we looked at the variants now from the literature and we curate them and put them into databases. So there's a lot of things already happening for the GRIN genes, which will make things way easier to make a decision what the function of a variant is, because people did a lot of molecular work already. However, for the majority of the GRIA genes, there's the patient variants which have been found, and the ones which will be found, there's no functional testing. So we have already started for the GRIN genes to develop like bioinformatic models which can use the functional data which has already been generated, and also some of the clinical data, to generate, models which can predict the function of a variant which has not been functionally tested. So it's similar, when you just think about it, how a car drives. If you have seen 100 cars stopping to drive and crashing to the right because the right front tire has a hole, then you can expect that another, different hole in the front tire on the right side of a car would probably also lead the car to crash on the right side, because the position where the mutation is plays a lot of role in terms of function. And there you can add also some other information. So, and we learned this already for the GRIN genes, not perfect, but we are actively every day working on this to make it better. So, and the interesting thing is, if you think about this car example, which I just gave, you can think about the NMDA receptor that would be like let's say a Ford, let's say just like as a type of car. And you could think about that the other GRIA genes, they build similar type of cars. For example, the AMPA receptor could be considered as a Toyota. So this is not the exact same car, but in general, they all have 4 wheels and they have a steering wheel, and the technology, how the engine works, is somewhat similar. So, and we're hoping now to develop with machine learning and a lot of biological information models where you can take all the functional experiments which we have generated over the years from the GRIN genes to then develop models which can also then predict the function in the other glutamate receptors, or the other GRI genes. And this is the whole project is about. From a biological perspective or biologist perspective, this makes sense because really an AMPA receptor, for example, is really similar to an NMDA receptor from the biophysical, so it's how it looks basically, but it's not the same. So, and this is what we have to do research on the next 2 years to make this really work. And the beautiful thing is these kind of machine learning models getting better and better and better with more advanced algorithms, but also with more and more data. And the beautiful thing is like through meetings like this where scientists and families come together, where everyone is arguing, hey, we need to share data, we're getting more and more data. And so that's why we are working on this, and we are very hopeful that we have a relatively good predictor by the end of this year for the GRIN genes. And we have already seen the poster for Ludovica. And we did also some proof of principle already for moving this information to the GRIA genes, and particularly the AMPA receptor. And we are very hopeful that this project could be successful, and we're working very hard on this. And we're very thankful for the support. Thank you.
Okay, thanks very much to Dr. Kalev, Dr. Coombs, Dr. Lal, and Dr. Bowie for your talks today. And just a couple of quick things to mention. I know we're already over time, but when we took in all of the applications for funding, there were 3 that didn't rise to the same level that we have let know that we can't move forward with funding them. But there's still another 4 that we've received that we would love to be able to fund. We think it would be really valuable research moving forward. And we've received 3 more that we haven't even reviewed yet that look pretty promising. So there's definitely no shortage of great science that could be done if we have the resources to do it. So so there's definitely a lot of opportunity, and we're excited about that. Last thing I'll say is that with regard to the plan that we presented before, so I mentioned that that's a draft. And so we have a board meeting next week, and then we have another one towards the end of the month where we're gonna be finalizing this over the next 3 weeks. If anyone has any feedback or questions, definitely welcome that. You can email me, keith@curegrin.org, or Heather@curegrin.org. Thank you for coming.